Abacavir falls under a class of anti-HIV drugs known as nucleoside reverse transcriptase inhibitors, abbreviated as NRTIs. To understand how abacavir treats HIV, let's first re-explore what happens in our cells once HIV has infected and entered our cells. Remember that HIV is an RNA retrovirus. The RNA means that instead of being double-stranded like our normal DNA, the HIV genetic code consists only of one strand. The retrovirus means that the HIV RNA requires an enzyme to complete the other half of the viral RNA to form the proviral DNA. This proviral DNA is what hijacks our host cell DNA and creates the blueprint for the rapid proliferation of HIV. The enzyme required to convert the HIV RNA to the proviral DNA is called reverse transcriptase. Reverse transcriptase is included in the viral RNA package that was delivered into the cell. Let's look at how reverse transcriptase works to create the proviral DNA. Reverse transcriptase goes along the single-stranded RNA and adds the complementary half to form the double-stranded proviral DNA. Taking a closer look at this process, let's imagine that RNA represents one half of a ladder with half-completed runs. Reverse transcriptase completes the other half of the ladder by stopping at each run in the RNA ladder and adding the complementary run to form the full DNA ladder. The complementary runs that are being added to the RNA ladder are called DNA nucleosides, and they are naturally occurring. Abacavir treats HIV by halting this ladder construction process. Specifically, abacavir competes with the naturally occurring DNA nucleosides and forces the reverse transcriptase to incorporate abacavir into the ladder. Once abacavir is inserted and incorporated into the ladder, this then prevents the incorporation of the naturally occurring DNA nucleosides in the viral DNA ladder. In fact, DNA nucleosides can no longer be added to the viral DNA ladder, period, and thus the process of completing the ladder is terminated and proviral DNA is unformed. Thus, the viral RNA is blocked from being converted to the proviral DNA that would have hijacked our host cell DNA and caused the rapid creation of HIV virus. Let's take a look back now at our host cell. The action of abacavir blocks the completion of the conversion of the viral RNA to the proviral DNA. Thus, HIV cannot hijack our host cell DNA and insert the instructions for HIV into our blueprint. This helps control our viral load, or the amount of HIV in our blood.